Welcome to the review for regulation in prokaryotes. Question 1. Operons encoding the enzymes of catabolic pathways are generally blank, and those that encode for enzymes involved in anabolism are blank. So these are some terms that you may need to know. Catabolic pathway is one which is involved in breakdown. So, for example, the lac operon, because that breaks down lactose into its subunits. And anabolic is concerned with biosynthesis. For example, tryptophan, which synthesizes the amino acid tryptophan. Okay, so we know that enzymes of a catabolic pathway, a breakdown pathway, these are generally inducible, such as the lac operon, where the lactose is the inducer, really allolactose. And those that are involved in anabolism or biosynthesis are generally repressible, just like the trip operon, where it's only repressed when there's an excess of tryptophan that binds to the repressor protein. So really the reason for this is that catabolic pathways need to adjust to the amount of substrate. Okay, a substrate for a breakdown chemical reaction. So a catabolic pathways, they're generally always off until they need to be on, right? Because why make enzymes to break down a certain substrate if you don't even have that substrate around? It's just a waste of energy. But anabolic pathways, they need to adjust to the levels of product. Okay, so for example, tryptophan is the end product of a biosynthetic pathway. Now, it's usually always on because most products of biosynthesis, a cell needs to function. So these operons are always on so that you make the product that you need until there's just too much of the product. And once there's enough of the product or even an excess, there's no reason to have the operon on anymore. So therefore it's on until it's repressed by high levels of whatever you have. Problem 2. For the following diploids, predict whether lactose permanase will be transcribed in the presence of lactose or in the absence of lactose. So it's the only structural gene they gave us, but remember that LAC-Y gene is the one that encodes for lactose permanase. Okay, so let's look at the diploid in part A. You have, on one copy, you have a normal LAC-I, which encodes for the repressor, a normal functioning promoter, and then a non-functional LAC-Y gene. On the other copy, you have a non-functional repressor gene, a functional promoter, and a functional LAC-Y gene. So let's look first at the repressor. You have one functional copy, wild-type copy of the repressor protein. So one functional repressor. Remember that the repressor is a protein, so therefore it is a trans-acting element. Okay, both promoters are wild type and they work fine, so that's no problem there. And then you do have one functional copy of LAC-Y. Okay, so even though the functional copy of LAC-Y and the functional copy of the repressor protein are on different copies of the operon, because the repressor is a transacting element, it's going to make up for the mutated repressor protein on the second copy, and this will respond normally, okay? It will have a normal lac operon phenotype because the wild-type copy of the repressor makes that happen. It restores this normal lac operon phenotype. And the normal phenotype for here is that LAC-Y, lactose permanase, will only be expressed in the presence of lactose, when it is induced by lactose. Okay, so the first option, yes, it will be expressed in the presence of lactose, and it will not be expressed in the absence of lactose. For part B, you have 0 copies of a functional repressor protein. So that right off the bat should tell you that you will not have a normal wild-type phenotype for the lactoperon. Okay, you have 1 functional promoter. And this promoter, remember that the promoter is because it's actually a part of the DNA sequence, is cis-acting. So this functional promoter is on the same copy of the operon with a non-functional LAC-Y. So this is with LAC-Y minus. And then you have 1 non-functional promoter, which again is cis. And this is with a normal copy of LAC-Y. Okay. So there's no repressor protein. So if everything else was fine, if the promoters were fine, if the structural genes were fine, if there was no repressor protein, that means it would always be on because there'd be nothing to repress it, right? But, however, that's not really what matters here. What matters here is that there's only 1 functional promoter. So this functional promoter, P+, is on the same operon as Y-. So even though this promoter works, this structural gene will not make a functional product. So the first copy will make no lactose permease because it doesn't have the correct gene for it. And the second copy has a correct gene for it, has the wild-type LAC-Y, but then it has a non-functional promoter. So this one will not make it either. So no lactose permease on either copy of the operon. Okay, and that, remember, that's because there's a huge difference between transacting and cis-acting. If a cis-acting promoter is non-functional, then there's no way for RNA polymerase to recognize the operon and start transcribing. So the structural genes downstream of that non-functional promoter will never be expressed. Okay, so you have no lactose permease from either copy, so lactose permease will never be expressed. Either in the presence of lactose or in the absence of lactose. Okay, so just to drill this in, that you need to know what the transacting elements are and what the cis-acting elements are. So just to recap, if you're making a protein like a repressor protein, those are normally transacting because they can diffuse to whichever copy of the operon. But cis-acting elements are usually the operators, the promoters, and the structural genes because they are part of the DNA of the operon. Problem 3. Complete possible genotypes for the diploids below. A check indicates lactose permease is expressed. 0 means it is not expressed. So let's look at this first diploid. If you see on the right, lactose permease is expressed regardless of lactose levels. Even if it's present or absent, it is expressed. So the phenotype for A is always expressed, or always, the operon is always on. And let's just take stock of what we have. So we have a wild-type copy of lactose permease gene, right? We have a constitutive operator, which means this is an operator mutant that cannot bind the repressor, so it will always be free, it will never be blocked. Then you have a non-functional promoter here. And then you have a functional promoter here. Now remember that promoters are cis-acting because they are part of the DNA of the operon. So even though you have a wild-type copy of lactose permease gene, this copy is actually never going to be expressed because the promoter is non-functional and therefore cannot bind RNA polymerase. So let me draw our blanks here. Well, I'll draw them in red actually. Oops. Okay, so because that the first copy has a non-functional promoter, that means that even though you have a wild-type copy of LAC-Y, that really doesn't matter because it will never be expressed. So the blank on the second copy, that must mean that you must have a functional LAC-Y gene again, because that is the one with the normal promoter. So you definitely need to have wild-type LAC-Y. So we'll draw that as LAC-Y plus. So that is one blank. Then we need the operator for that, and then we also need to talk about the repressor genes for both copies. Okay, so if we have a wild-type promoter, that means that this LAC-Y functional gene can be expressed. The operator needs to be functional too, so you can't have a mutant operator. Well, actually the operator could be either one. So it could either be O plus or a mutant O constitutive, because remember, the O constitutive can't bind repressor either way. So let's take it one at a time, because there's actually a few answers. So we know for sure that on this second copy, you need to have a functional LAC-Y gene. So let's say if the second copy we have LAC-Y wild type, and let's go with a normal operator right now. If we have a normal operator, normal promoter. A normal operator can respond to levels of lactose, right? So if there's no lactose present, the repressor protein will be bound to that operator and it will block transcription. But we know that they're telling us that the phenotype here is that this permease is always expressed. So even though this operator can bind the repressor, it never is, because as you can see, the lactose permease is always expressed. So that must mean, if we have a normal operator, that must mean that both copies of the repressor gene are non-functional. Okay? So this is one answer. So if there's no functional repressor gene, that means you won't make a repressor protein, and therefore even though you have an operator that could bind it, it never will because there's no repressor protein to bind. Okay, so this is the one answer. But, as I said before, you could have a different answer if you have a mutant operator. So again, we're going to need the LAC-Y functional gene. And then if you have an operator OC, that means this operator can never bind a repressor protein, right? So you have a normal promoter. And if it can never bind the repressor, then it could either be LAC-I wild type or LAC-I mutant. And the same thing for here. It doesn't matter if you have a functional repressor or not, because even if it's functional, this operator won't be able to bind, and you can always make lactose permease. Okay, so these are the two different answers for part A. For diploid B, if you look at the phenotype, you see that lactose permease is expressed when lactose is present, but isn't expressed when lactose is absent. So this is a normal wild-type phenotype for the LAC operon, where it can respond to different levels of lactose in the cell. So what do we have here? We have a non-functional repressor protein, LAC-I, minus. We have a normal promoter, normal operator, and then on the second copy you have a non-functional LAC-Y, lactose permanase gene. So we know that in some cases when lactose is present, you are making lactose permease. So this other copy must have the wild-type copy of LAC-Y. So I'll draw that here, LAC-Y plus. Okay, and the promoter and the operator on that actual copy of the operon is fine. You want to take a look at that because remember the promoter and the operator are cis-acting, so that's fine. But you know that there's a normal phenotype, which must mean that there's a normal repressor copy in the second diploid, in the second copy. Or else, if there was no wild-type copy of the repressor protein, then there would be no selection between whether lactose was present or absent. It wouldn't be able to regulate. Okay, and really, so this is transacting. So it could go and bind to the operator here to regulate this LAC-Y gene. But the promoter and the operator on these could either be wild type or mutant. Because again, it doesn't really matter because this is a non-functional copy of lactose permease, so it's not really going to affect the phenotype that you see. So definitely you would need a wild-type copy of the repressor protein, LAC-I+, and you would definitely need a wild-type copy of lactose permease, LAC-Y+. Problem 4. Of the following choices, which will produce beta-galactoside transacetylase even in the absence of lactose? So this is a non-wild type phenotype for the lac operon, right? Because a wild type phenotype would be where the LAC-A product, beta-galactoside transacetylase, is only expressed in the presence of lactose. So we're looking for something a little bit different. So let's take it one by one. So part A, this genotype has LAC-I minus, so no repressor protein. So far that bodes well for the phenotype we're looking for. It has a non-functional promoter. A constitutive operator, which means that that operator cannot bind repressor even if there was one, and a wild type LAC-A. So 3 out of the 4 elements here work toward making us think that this is the right answer. So you have a normal LAC-A product, which is what we want because we want it expressed. You don't have any repressor, which is good because that means the operon will always be on, even in the absence of lactose. You have a constitutive operator, which means that even if there was a repressor protein, the operon will still always be on. But the problem here is the non-functional promoter. So because of this, and this is a cis-acting product, so even if you put a different copy of the operon in there, it wouldn't fix it because this promoter is cis-acting, which means that this LAC-A product will never be expressed. So A is not our answer. Okay. B has a normal repressor, functional promoter, functional operator, and a wild type LAC-A. So this is what a normal LAC operon looks like. You have wild type repressor protein that can make repressor that will sense the levels of lactose in the cell. You have a normal promoter, so RNA polymerase can recognize the operon and start transcribing. You have a normal operator, which will bind the repressor, and you have a functional structural gene. So this will give you the wild type phenotype of the lac operon, which is not what we're looking for. Remember, we're looking for something that expresses LAC-A even when there's no lactose around. Okay? C has a repressor protein, a normal promoter, operator constitutive, which is good because that, even if there is repressor around, which in this case there would be, it will never bind it. So the operator will always be free and won't bind transcription. But in this case, you have LAC-A minus. So there'll be no functional beta-galactoside transacetylase. So even in the presence or absence of lactose, this won't be our answer. So finally, part D, choice D should be the answer, but let's just double check. So there's no repressor, so that's good for us. There's a normal promoter, normal operator, and a wild type copy of LAC-A. So this works well because there's no repressor protein, so therefore the operator will always be free. RNA polymerase can recognize this promoter because it's wild type, and it will begin to transcribe, and there's a wild type LAC-A gene, so you will make beta-galactoside transacetylase whether there is lactose in the cell or not. So choice D is the answer. Okay, question 5 and 6 are just some simple fill in the blanks. So when cellular levels of tryptophan are scarce or low, the TRPR repressor protein is inactive, right? Because it's made in its inactive form, and it's only activated when bound to the co-repressor tryptophan. So in its inactive state, it cannot bind to the operator. So just a quick tip that repressors bind to the operator only when active. And it is RNA polymerase that binds to the promoter. So when tryptophan levels are low, the TRPR repressor is in its natural state, which is inactive, and therefore cannot bind to the operator, and transcription continues. Question 6, that when cellular levels of lactose are scarce or low, the LAC-I repressor is in its active form, which means that it can bind the operator. So this is the difference between the LAC operon and the TRP operon. For the LAC operon, which is inducible, the repressor protein in its natural state is active and bound to the operator to block transcription. But when the operon needs to be turned on, the inducer, which in this case would be allolactose, binds to the repressor, inactivating it so that it can no longer bind and block the operator. But in the TRP operon, the normal repressor is inactive because under normal conditions, the TRP operon is being expressed, but when there's enough tryptophan in the cell, the tryptophan can bind to the repressor and activate it so that it can block and turn off transcription of the operator. Problem 7. Jay, the geneticist, is working with a strain of E. coli that seems to have an abnormal LAC phenotype and needs help figuring out which mutation is causing it. He has narrowed it down to 2 possible LAC mutations that present with the same phenotype, either LAC-I-, a defective repressor that cannot bind to the operator, and LAC-O constitutive, a defective operator that cannot bind the repressor. Which phenotype must Jay be observing? Design an experiment to test which mutation is present and give a set of guidelines for Jay to distinguish between the two. So let's look first and figure out what phenotype Jay must be observing. So he knows that the mutation is either that there's a non-functional repressor or there's a mutant operator that cannot bind a functional repressor. So if there's no repressor operator binding, that means that transcription is never going to be blocked, and therefore the phenotype he must be seeing is that the LAC operon is on, or that the LAC structural genes are always expressed regardless of lactose concentration. Okay, and that's because either way, there's no way for a repressor to bind to the operator to block transcription. So now we have to figure out an experiment for him to figure out which of the two this mutation really is, because if a phenotype is the same, there's no way to see which one it is, just by observing the phenotype. So remember from earlier problems in this chapter, in the previous chapter we did with tryptophan, and then a few chapters ago we did with lactose, what's known as a merodiploid. So we're going to exploit that. So remember that merodiploids can be constructed by adding a second copy of either a full operon or just some of the genomic genes via a plasmid. Okay, so we're going to introduce a plasmid to see if we can figure out which mutation the genomic operon has. Okay, so let's go over the genomic one that we have. Okay, so we either know that it's a mutation in LAC-I, or it's a mutation in the operator, and we don't know which one has the mutation and which one doesn't. But then we have some functional structural genes, because he's seeing them being expressed even if lactose isn't present. Okay? So if we're going to introduce another LAC operon, a copy of it, via a plasmid, we want to have some way to distinguish between the two. So what we're going to do in this genomic copy is mutate the LAC-Z so that it no longer makes a functional product. Okay? So let's just assume that he has all these techniques that we need at his disposal. So the first thing in our experiment, we're going to mutate the genomic copy of LAC-Z. Okay, so let me just put a dash there so we know that that is no longer functional. And this way, when we want to see if either the genomic copy or the plasmid copy is being expressed, if the genomic copy is being expressed, we won't see any LAC-Z. Okay, so that's a good way to distinguish. Now, what we need to do is we need to construct a plasmid with a specific genotype that we want. So, the plasmid genotype that we want is going to have functional LAC-Z, so that it's distinguishable from the genomic copy, but then we want non-functional LAC-Y, non-functional LAC-A. So that takes care of the structural genes on this second copy of the operator. Okay, but we want to have a wild-type repressor gene and a wild-type operator. So we want to have LAC-I plus and a wild-type O plus operator. Okay, so the reason why we have a functional copy of the LAC-I gene, a wild-type copy, is that now this plasmid will make a functional repressor. And this will be a functional operator that can respond to repressor. Okay, so one, you have to mutate the genomic copy of LAC-Z, and then two, you have to construct this plasmid. And then obviously you have to introduce the plasmid and then take observations of the cell either in, well, both with low lactose supply and then again with high lactose supply. So what are the guidelines that we're going to tell Jay to look for? Okay, if the original genomic mutant was LAC-I, then the functional LAC-I plus repressor that we added in should restore the normal phenotype of the LAC operon. So if the genomic was LAC-I minus, the plasmid will restore a normal phenotype. Which is where the structural genes are only expressed when lactose levels are high. Okay, so for the guidelines, you want to check that under low lactose, no structural genes are expressed. And under high lactose and only lactose, if there's no other energy source around, you want to check that all structural genes are expressed. Okay, so even though we mutated some of the structural genes, because LAC-I is a transacting element, the repressor is transacting. If lactose levels are high, and the repressor protein will be inactivated by allolactose, that means that it can go and express. So if the genomic copy was LAC-I minus, that means that its operator was normal. Okay, so if genomic copy is LAC-I minus and then also normal operator, that means that the transacting repressor protein can go ahead and act on either copy of the operon. Okay, however now, if the genomic copy had a normal LAC-I plus, but then had the constitutive operator, this will have some different guidelines. So if the mutant genome was LAC-OC, and remember it's only one or the other, he's narrowed it down to the two, and he's just trying to distinguish which one it is, then only the genes on the genome copy will always be expressed. Then only genomic structural genes will always be expressed. And that makes sense, right? If there's two normal copies of the repressor protein gene, then only the one that doesn't have a functional operator will always be expressed because it'll never be blocked by the repressor protein. So the guideline for this, you want to check that under low lactose levels, which is normally when the operon is repressed, you want to check that the genomic structural genes are being expressed. So you want to check for levels under low lactose, check for expression of LAC-Y and LAC-A. Okay, so these are his guidelines. If he sees that under low lactose levels, none of the structural genes are expressed, then that means that the original genomic mutation was LAC-I minus with a normal operator. But if he sees that under low lactose levels that LAC-A and LAC-Y are expressed, that means that the genomic mutant was LAC-I plus with normal repressor, and that has the mutant constitutive operator. So this is how we can help him see which mutation is actually causing this phenotype. Determine if the following statements are true or false. The LAC operon is an anabolic operon, meaning it encodes for enzymes involved in a degradation pathway. Okay, so while the LAC operon does encode for enzymes that are responsible for degrading lactose and breaking it down, this is actually an example of a catabolic pathway. Okay, so that makes it false. If it said catabolic, that means that it would be true. An anabolic operon actually encodes for enzymes that are used in synthetic reactions, like biosynthesis of tryptophan. 2. Inducers cause conformational changes to repressor proteins, which render them unable to bind to the operator. So this is true. And this is how inducers turn on operators, by effectively shutting off the repressor. On the other hand, a co-repressor causes conformational change to repressor proteins, which lets them bind to the operator, which otherwise they wouldn't be able to. So it's slightly different. So inducers cause the conformational change that makes the repressors inactive, and co-repressors make a conformational change that makes the repressor active. 3. Lactose is a polysaccharide made up of 2 units of glucose and 2 units of galactose. This is false. Lactose is a disaccharide, and it's made of 1 unit of glucose and 1 unit of galactose. So it's a disaccharide. 4. When neither glucose nor lactose is present, transcription occurs. And they mean here transcription of the LAC operon. So this is false. In order for transcription of the LAC operon to occur, the repressor protein must be bound to an inducer, allolactose. If no lactose is present, then an inducer isn't present, and therefore the repressor is still bound to the operator to block transcription. 5. When both glucose and lactose are present, transcription of the LAC operon is turned off. So this is true. And that is because even though lactose is present, if there is an alternative preferred energy source like glucose, the LAC operon isn't going to be turned on. Okay, so that is because when glucose is high, cyclic AMP is low. And when cyclic AMP is low, that means that transcription is not going to occur. Okay. 6. The LAC repressor is a trans-acting element, where the TRP attenuator is a cis-acting element. So this is true. Remember, because the LAC repressor is a protein which can diffuse to any copy of the LAC operon that is in the cell. However, the attenuator in the TRP operon is that region 4 of the leader sequence, and that is a piece of mRNA. So that is part of the mRNA of the whole operon that's getting transcribed, and therefore it can only act on that operon that it is a part of. 7. The tryptophan is a co-repressor of the TRP operon, and this is also true. When excess tryptophan levels are in the cell, it binds the repressor protein, which activates it and therefore turns off transcription of the operator. So we call tryptophan a co-repressor. And then finally, the TRP operon has 5 structural genes encoding 5 different enzymes required for the synthesis of tryptophan. So this is false. There are, in fact, 5 different genes, but these genes only encode for a total of 3 enzymes. And that's because 4 out of the 5 genes only encode for subunits of proteins. So once all 5 genes are translated, 4 out of the 5 of them, I think it's 2 of them form together to form one enzyme, and then another 2 form to form a different enzyme. So altogether you get 3 enzymes that are in the biosynthesis pathway to make tryptophan. Okay, so this includes the review on prokaryotic regulation. We talked about the operon concept, the lac operon, which was inducible, the TRP operon, which was repressible but also had that second tier attenuation. So I hope this was helpful. I would really suggest you go over the notes, make sure you understand cis and trans and acting elements, and also know for each operon what part, which element of the operon does what in the process.